Welcome, everyone. My name is Diana Wall, and I'm the Science Chair of the Global Soil Biodiversity Initiative. It's a real pleasure to have you all with us today when we talk about soil biodiversity and agricultural sustainability. This is going to be a really good panel, but I have first some housekeeping news for you. Please use the question and answers. The question and answers we'll be monitoring those, but we will not write answers to you because this is all going to be recorded and it's going to be on YouTube. And so the answers won't show up, but please answer in Q&A only, not in chat or anything else. The other thing that I want to do is just make sure that to make the questions come throughout the whole webinar. Please do keep looking back and see if your question hasn't been answered. You may want to re-enter it. So now what I'd like to do is introduce our moderator, Johan Six. Johan is from ETH in Zurich. Johan, welcome.
Thank you, Diana. So welcome to all of you. And I will just quickly share my slides. So we have today quite an interesting panel. We have quite a bit of caliber in our panel. And so I would like to just shortly introduce them. And then I will also just give a short introduction to the whole webinar. So the first person on our panel is Gerlinde de Deyn, and she should be showing up. Yes, there is Gerlinde. And so she is a professor of soil ecology at Wageningen University. The focus of her research is on plant-soil interactions and feedbacks in which she links soil and plant ecology and remote sensing, with the aim to achieve higher nutrient use efficiency and produce more nutritious crops, reduce greenhouse gas emissions from soil, and suppress the buildup of pests and diseases. So quite a mouthful, but she will indeed give more explanations on that in her presentation. The second person we have is Sara Moreno. And Sara Moreno is at the National Center for Agriculture and Food Research and Technology, and that's located in Madrid, Spain. And she really focuses on soil nematodes in agriculture, soil functioning, and management strategies to preserve soil diversity. And so again, Sara will also give some further explanations on that. The third person is Jonathan Leake, and you will see there is Jonathan, and he is at the University of Sheffield as a professor of plant-soil interactions. He has worked there for quite some time on plant-soil microbial interactions, mycorrhizal functioning, and more recently has also led some bigger consortiums on UK arable farms, investigating the methods of sustainable agriculture and especially then regenerative agriculture. And there he has a particular interest in the effects of leaves on soil biology and functioning. Jonathan is also a scientific advisor to the Sustainable Soils Alliance and has considerable experience communicating soil science to farmers and policymakers. So I'm sure you're also looking forward to hearing from him about LACE and how to communicate about that with farmers and policymakers. And then last but not least, we have Stuart Grandy that's also joining us today. And you should see Jonathan coming on soon enough. And he is a professor of soil fertility and co-director of the Center of Soil Biogeochemistry and Microbial Ecology. Stuart has mostly contributed to the optimization of nutrient and organic matter management in agroecosystems by examining interactions between plants, microbes, and minerals. And also he will give some more details on that in his short presentation. So myself, just to give a bit more background, you know, I do work here at ETH Zurich as a professor, and I basically lead the group of sustainable agroecosystems. And so obviously that also explains why we're today talking about soil biodiversity and effectively sustainable agriculture. And I've given a bit of task to all of our panel members to indeed, you know, not talk too much in theory about this, but effectively come up with some examples and practices of this. Because I think we indeed know, you know, quite well, you know, what microbes and our soil biota can do in general. You know, they are obviously very important for nutrient cycling. They are important for degradation of pollutants. They can also help obviously with plant growth and stress tolerance. There's a lot of talk today about how they can maybe help with climate regulation, you know, by regulating methane and CO2 and N2O emissions. And then also, you know, more and more, it's also evident that they can play a role in pest and disease control, both below ground and above ground. And so, you know, those things are quite well known, but some of the details clearly not. And then I think especially, you know, we know already that agriculture can have quite a bit of an influence on that, because you know, lots of the industrial agricultural practices are effectively degrading our soils and therefore affecting our soil biota. And then the question is indeed, what can some of these alternative practices in agriculture do to actually make it again a healthy soil and a soil that is really helping maintaining the soil biota in their functioning. And so there's actually quite a bit of research on that too. And obviously it has a lot to do with you know, how we're managing our soil directly and how much we can keep the structure and the connectivity going. But then with the hope, and that's where I think indeed, you know, quite often it's then just assumed if we have a better soil, that indeed is enriched biodiversity can help with nutrient cycling again, doing methane oxidation, having lower nutrient leaching. And so that's where we today will then be talking more and giving some examples of how this could go. So with that, I would like to open the floor to Gerlinde, and so that she can give her short introduction.
Thanks a lot for the introduction and for the invitation, of course. And indeed, I'm happy to take on this challenge to present very briefly what my research is about, and also to actually address the three short questions that were posed to us. I hope I can capture them in 5 minutes, but basically this is just the onset for then the discussion later on. So what do I really do in terms of this soil biodiversity and sustainability? Well, I'm mostly interested in this functioning of this soil, and mostly from a community perspective. And even more so specifically, how do plants interact with the soil and how does it then feed back to plant growth, but also to reduced greenhouse gas emissions and other soil-based functions. And one of the tangible examples here is how cover crops create a legacy to subsequent main crops. And this is just an example of how we played around with different monocultures and mixtures. And here we can clearly see on the left-hand side, this is a legacy of a mixture of 2 species, which actually was worse than the average of the monocultures. While on the right-hand side, we get this legacy that was better than the average of the monocultures of these 2 species. And so the way it works is indeed via nutrient cycling effects, but actually it's because of the quantity and quality of the amount that goes in via above and below ground that has knock-on effects on this soil community that changes in activity and in size. So that's very relevant. And actually, we also made use of drones there to study that in more detail. We also study effects of resilience and to drought and flooding in relation to management of agricultural systems, but I can't go into detail there. And my work also gets more and more connected to this idea of circular agriculture. And what do we mean with that? Well, here is a scheme on the right-hand side. I hope you can see that fully, because for me, there's a bit of overlap of the Zoom layout. But basically what it illustrates is that indeed we harvest from the soil and we produce food with that. And the idea is that we actually should also return as much as possible back to the soil and keep the system as close as possible in terms of nutrient use and also in terms of energy use efficiency. And hopefully also, on the one hand, combat climate change with that, so reduced greenhouse gas emissions, but also even promote diversity. Because of this impact of managing our soils, we should reduce impact on the more natural environment as well. And indeed, the soil is the basis here. That's the idea. And that's also a role for us, I think, to stress this. Because the idea is that a lot of these new kind of returns come back to the soil. And one example is like animal manure, but we have to be careful there that these manures can also have antibiotic residues, for example. So that's one of the studies we do is checking this impact of different types of antibiotic residues in manure on this knock-on effects on the functioning of the whole soil microbial community and the emissions of greenhouse gases and so on. Next to that, you see this connection also to natural systems, because there we have the issue of eutrophication, which is suppressing diversity. So what we do there is make use of harvested plant material from there, export it from the natural system and bring it back to the arable system. So that's in different ways like composting, bokashi, and different forms, with the idea that we will build up or restore soils in the arable systems and promote biodiversity in these more natural systems again. And we even connect there with aquatic systems where we make use of invasive aquatic plants. And that's a study we do in Zambia. So just to show that it becomes more wide in a way, more connected what we're studying in terms of soil in agricultural systems, just because we're so interconnected by now. And in the end, that even feeds back to healthy food. So it really becomes relatively big. Then what are the challenges? And I'm just listing a few here because of time. What I think are still pretty big challenges, or still this variation that we have to deal with in terms of biodiversity in space and time. This connection between really the taxonomy and the functioning, and ideally go really more mechanistically still and go in situ. We know a lot of things in the lab, but still these dynamics are happening in the fields. And to give advice to farmers, they have to manage their fields, right? What can we do to overcome these challenges? Well, we still need experiments, I think, to be able to disentangle causes and consequences. Have indeed these nice databases on how taxonomy and traits characteristics are really coupled. And if we really understand, we should be able to model it as well, I think. And modeling also helps us to really zoom in and out at the right level to know what do we really need to know. And I think in practice, to connect also to policymakers, indicators can be very helpful. And for in situ measurements, I think sensors can be of good use. Some sensors are already available, others may still need to be developed for fitting our own purpose. Then I come to my final slide. That was the question on how can we inform policy? How does our work contribute to policy? I think in the first place, it is providing the scientific basis for these reports produced by IPBES and IPCC, and also give advice to more local policy. Like sometimes we do get these questions, like, for example, the impact of solar parks on soil quality, on arable fields even. And next to that, I think, yeah, we really need to work very multidisciplinary. And again, these soil indicators, I think, can be a good communication component, and also something that can be used to really track where we are in the trajectory to where we want to get. And related to that, in this publication last year, which was part of a special issue on soils and nature contributions to people, we made this scheme. It's a little bit a busy scheme, but just to illustrate, I think indicators can indeed be a very handy tool, but you do need to connect them together with the stakeholders, together with the policymakers, so that you do speak the same language and that it is really science-based. And from the science perspective, that's what we have on the bottom side, all these different pools, let's say, of information that we have. So soil biology is definitely a part of it, but it's definitely not the only part of it. And we do need to have this integrative approach to really get to change, basically. And for sure, we can't do it on our own. But given the current situation and technologies and the communication, we can, I think, achieve a great deal in the next couple of years or the next decade. And with that, I'm happy to stop sharing.
Thank you, Gerlinde. And so I just want to remind the audience that you can indeed start putting questions into the question and answers. And if I could indeed ask now Sara to share and her expertise.
Thank you very much, Johan, and thank you very much, Gerlinde. Let me take this out. So the title of my talk today is Soil Food Webs and Agricultural Sustainability. I do work with soil nematodes. As many of you know, soil nematodes is a group of soil animals which is extremely abundant and extremely diverse. A recent paper showed that nematodes in soil, nematode abundances in soils, range from a few hundred to several thousand nematodes per 100 grams of soil. That makes nematodes probably some of the most abundant animals on Earth. And they are not alone, because other soil animals are extremely abundant too, such as mites, collembola, and so on. Some estimates say that 50% of animal biomass on Earth is underground. So my research lines deal with how do soil animals favor sustainability. Soil animals interact in extremely complex soil food webs, and they interact through trophic and non-trophic relationships with one another and with other components of the soil biota. For example, many nematodes and many other soil animals are bacterivores and fungivores, and they feed on bacteria and fungi, and they regulate microbial populations and nutrient cycling, increasing soil fertility. Previous research showed, for example, that crop biomass is higher in soils with nematodes than in soils in which nematodes have been experimentally excluded. We have also found that some nematodes, those bacterivore nematodes closely related to microbial communities in the soils, are very related to grain yields in agricultural systems. Other soil animals are predators and omnivores, and they control harmful organisms, inducing soil suppressiveness. Soil suppressiveness is the ability of soils to fight against diseases and pests. And we have also worked on studying how these predatory and omnivore animals in the soil exert as pest control agents. But of course, not all soil animals are beneficial to crop yields or crop performance, and many of them are crop pests or they induce crop diseases. Actually, more than 4,000 nematode species are plant parasitic, and they cause losses of $125 billion a year. So my research line deals with how soil food webs relate to soil organic carbon and other soil nutrients and crop performance. In general, how soil food webs deliver ecosystem services, such as yield, fertility, and pest control. These relationships here are threatened by climate change factors, such as warming and drought, and we studied too how different types of soil management may act as mitigation strategies against climate change. Those mitigation strategies are, for example, minimizing soil disturbance. We've clearly seen how reducing tillage, for example, using reduced tillage or no tillage, increase the participation of those bacteriivore animals in the soil, mineralizing nutrients and increasing soil fertility. The second strategy would be diversifying cropping systems. It is well known, it has been shown everywhere, that including rotations in agricultural systems reduce the amount of pests in the soil. For example, reducing the abundances of plant parasitic nematodes. And finally, increasing organic matter inputs, especially in the soils in which I work, with are very poor soils with low contents of organic matter in quite arid conditions. So we have seen that incorporating cover crops into the soil also optimize the participation of soil animals in nutrient cycling, increasing soil fertility. So in relation to the major challenges in this topic, I think the first one would be to identify soil animal diversity, joining morphometrical and molecular identifications. In second place, quantifying soil animals' contribution to ecosystem services. This, of course, has been done, but much work remains undone. And the third one, transmitting our findings to farmers and other stakeholders. I think it is very, very, very important to establish effective communication channels among farmers, scientists, and policymakers to transmit that protecting soil diversity and soil fauna is protecting agricultural sustainability worldwide. And that is all. Thank you very much.
Thank you very much, Sara. And if I could ask Jonathan to present.
So thank you very much for the invite. And I want to just talk to you a little bit about some of the work that we've been doing on biological regeneration of soil health. I mean, the opening photograph tells a big story, actually. It's just, we have a permanent arable field into which we introduce a 3-year ley. So it's grass and clover. It was just mown, and the soil radically changed over that 3-year period. The stopping tillage, the feeding of the soil with roots and nitrogen fixed by the plants was transformatory. And I'll come into the details of what it actually achieved shortly. And you can see that it's rebuilt some of the structures that you see in these long-term permanent hedge soils, which are decades, well, probably centuries old. So I suppose our starting point was actually looking at the extent to which intensive arable cropping was causing soil degradation. And the simple way of looking at this initially was to take a standard weight of soil and grow wheat in it, just watered with standard tap water, no nutrients added, and looking at what happened. And when we looked down into these soils, we could see straight away they were very different in structure and enormously different in function, as you can see from the response of the wheat plants. And the structural differences resulted in very major losses in water holding capacity associated with a very large loss in soil organic matter. But also, and you can see this very clearly here, the soil lost its structural stability with this loss of carbon. And so the larger aggregates, which are dominant in the better structured organic rich hedgerow soils and field margin soils, in these permanently arable soils, these are lost, and the major aggregate fraction that we see here actually is the smallest fraction, less than 53 microns. And there are consequences of these changes. This is a landscape scale effect that we see over much of Yorkshire, where these soils came from. And it's an area which is now subject quite often to extensive flooding that affects properties and livelihoods. And the soil is biologically depleted. It's not just structurally and chemically depleted. The earthworms are depleted, the mycorrhizal fungi are depleted, the organic matter is depleted, as I've already said, the structure is degraded, and we also have problems here of crop pests and diseases that are also connected to this loss of soil biota. What's been going on here is continuous arable cropping for decades, soil ploughed every single year, wheat being grown in 60% of the rotation. So we were interested in trying to reverse that. And so what we did was we put in lay strips into these permanent arable fields so we could compare alongside the same field, same soil types. All we did was locally change the management and started to investigate what the result of this was. And I suppose one of the first things to note is that this is when the wheat crop's been harvested off the arable field. And in the late summer, we've still got a bright green strip photosynthesizing, fixing carbon and supplying carbon into the soil. And other data suggests that actually these kinds of lay systems probably more than double the amount of carbon going in through roots into the soil. Feeding the soil with live roots is probably one of the key processes to support soil biology, actually. And it's a central pivotal role in regenerative agricultural systems. So this is what our system looked like, as I say, and then we also conducted more detailed experiments within this system. So we actually dug up blocks of soil, shoved them in a deep freezer to try and eliminate earthworms, or largely eliminate earthworms, put those blocks of soil back in the field, sow them with grass and clover, and then some of them we added earthworms to augment the earthworm population. So we had controls that we didn't freeze. And so the frozen samples got a few earthworms in over time, but not very many. The controls, which were not frozen, had a moderate amount of earthworms, and the ones that were frozen, and then we added more earthworms back in, we managed to boost the earthworm population. So this is work that Jamal Hallam and others have done. Some of this is published and some of it's not yet published. So looking at this, one of the key things we found was that the organic carbon in the greater than 2 millimeter aggregates was not affected by the deep burrowing, the INIA-CSIC earthworms. The numbers of those didn't make a difference, but the number of endogeic earthworms, those that burrow through the soil, really was significant in terms of its effect on soil carbon storage, and the same applied to nitrogen. So I suppose one of the key messages here is it's not just about the numbers of organisms, it's also about the types and the numbers of particular types of organisms, which start to become important. And sorry, this is a very busy slide, but to tell the whole story, these are the data I've just showed you in terms of earthworm numbers, and then it compared also to a 1-year lay in the field, enriched with earthworms. This is what happened by 2 years in the actual lay strips themselves, and this is by 3 years. And you see the massive increase in earthworm populations over this time. There was a positive feedback. Mycorrhizal fungi also, in terms of their colonization of wheat in field trials, also massively increased in the lay over a 3-year period, as did the diversity of mycorrhizal fungi increased. And of course, earthworms and mycorrhizal fungi are implicated in helping to build soil aggregates. And we see this big recovery in the larger aggregate size water stable aggregates in this system. So the lays, in this case, recovered to similar values of greater than 2 millimeter water stable aggregates as we had in the original hedgerow, undisturbed hedgerow soils. And what we then also see is that carbon is accumulating into these larger aggregate fractions. So here we have a biology driving structure resulting in protection of carbon and the accumulation of organic matter and the restoration of some of the properties associated with the soil as a result. So there's a strong positive feedback. Now, I highlight earthworms and mycorrhiza because we know mechanistically these are involved, but of course other organisms that we haven't tracked may also be involved in some of this. And these interactions, we think, lie at the heart of this kind of really substantial structural and biological and chemical alterations that we see and recovery that we see in this soil. Work that Emily Guest, a recently completed PhD student of mine with Thorin Helgeson at the University of York, they looked at the communities of mycorrhizal fungi in these systems, and they noted, for example, that the ley increased the diversity of mycorrhizal fungi, and in particular, the abundance of mycorrhizas relative to other fungi. So this is arbuscular mycorrhizal fungi. These are other groups of fungi. In the bacterial communities, actually, and these were averaged across 4 fields, there were no significant differences. There are different differences, but they're very subtle. But on the other hand, we did find differences in the amount of disease within the crops, and it was suppressed in the leys. And we know that these leys are developing disease suppressive microbiomes. We think bacteria are probably involved in this as well as fungi. And the upshot of this was when we ran trials with wheat grown on the soil out of these leys, what we found was really improved wheat growth, greater resilience to drought and flooding, and substantially boosted yields, no doubt as a result of the nitrogen inputs from the clover, the structural improvements, better water storage capacity, etc., etc. So coming back to these 2 core questions, one of the major challenges faced when studying soil biodiversity and sustainable agriculture and what needs to be done to overcome them, I think one of the major challenges is that clearly in time, trying to characterize which organisms are really important for sustainable agriculture, it clearly is the case that not all organisms are important to the same extent. And so identifying key indicator organisms, which are really informative and helpful, is quite important, I think, because these studies, for example, on bacteria showing no significant changes aren't really particularly informative. There must be other details in here that we perhaps haven't got a handle on enough, so there may be subcomponents of that. And interestingly, actually, we found more pathogens in the ley, but less pathogenesis in the plants that were grown in the ley. So there's anomalies that are quite difficult to understand, really. What needs to be done to overcome them? I think we do need to focus on trying to characterize more key indicators that are useful, that really provide good ways of monitoring improvements in biodiversity that lead to functional improvements. And how can this research lead to land use policy under climate change? Well, I think the sorts of research we're doing clearly indicates that if you change your management into practices that are beneficial for key groups of organisms, rebuild soil structure, rebuild soil carbon, and rebuild disease suppressive soils, you end up with better yields, you end up with better water holding capacity, you end up with better soil functionality. And so I think this does directly feed into policies. And in fact, the UK government is now providing financial support to farmers that are putting diverse leys back into their rotations on the basis of evidence such as that which we're providing from our own research. So I think there is hope as well that policymakers are actually responsive now to these issues. And that's my presentation finished. Thank you.
Thank you, Jonathan. If I could ask indeed, Stuart.
Thanks, Johan. It's awesome to be here. I'm looking forward to the panel discussion. Agricultural systems are nitrogen inefficient, leading to pollution of air, water, and the atmosphere. So today I'm going to talk about optimizing plant microbe-mineral interactions to support crop nitrogen requirements, focusing on interactions between mineral-associated organic matter, which I refer to as MAYOM, microbes and plants, and hopefully begin to suggest some ways that we might be able to better optimize nitrogen in agroecosystems, making it more efficient. So mineral-associated organic matter may be an overlooked but large source of nitrogen in agricultural soils. The majority of soil nitrogen is associated with mineral particles in agroecosystems. It's often 80% or more of the total N is in this MAYOM pool, which we operationally define as less than 53 microns. Whereas the POM has a very high C to N ratio, and it's often associated with nitrogen immobilization. This Mayon pool, this large pool, is mostly proteins and other nitrogen-rich molecules. And in the laboratory, it's often associated with nitrogen mineralization. And while some of these molecules are tightly bound to mineral surfaces, others are much more loosely associated and more available to plants and microbes. So how do we build Mayon, and what is the specific role of the microbial community? Previous work by Cynthia Kallenbach provides some insights into this, linking microbial physiological traits, crop rotational diversity, and the buildup of total soil organic carbon and Mayon, and also specific types of compounds in those pools. Here we have CUE, which is the carbon use efficiency of the microbes. And you can see on the far left here, this positive relationship between carbon use efficiency and soil organic carbon, going from a low crop complexity to a high crop complexity annual rotation. So as we get into a high crop complexity rotation, the microbial carbon use efficiency goes up, and the soil organic carbon in particular, we know it's the Mayon pool of carbon and nitrogen that's building in these systems. At the same time that we saw this increase in microbial carbon use efficiency in more complex rotations, we also saw this increase in the deposition of microbial lipids and microbial nitrogen-bearing compounds, in particular in this Mayon pool. This work was done at the Kellogg Biological Station, but in this new work by Maria Moshammer that's in press, we saw a very similar dynamic in Mead, Nebraska, where high temporal crop rotations changed the diversity of chemical inputs. This specifically led to microbial trait changes, including variation in enzyme expression, changes in the abundance of AMF fungi, and changes in specific bacterial biomarkers, which created a positive feedback on the soil organic matter pool. In particular, we saw more soil organic carbon in the Mayon pool and nitrogen, and this organic carbon and nitrogen possesses a very strong microbial signature. So how do plants access Mayon, and what is the role of the soil community therein? Andrea Yilling provided a really nice conceptual model of this, and she proposed that oxalic acid mobilizes metals, displacing Mayon and potentially stimulating microbial oxidase production. Glucose, on the other hand, in the rhizosphere produced by plants and microbes stimulates microbial enzyme production, and both of these separate pathways are capable of mobilizing Mayon and making it more available to plants and microbes in the rhizosphere. So Andrea followed up this work looking at these direct and indirect pathways of nutrient release from Mayon, and she added glucose and oxalic acid to soils from Illinois, and she saw that oxidative enzymes increased with the addition of oxalic acid, whereas these carbon acquiring enzymes increased with the addition of glucose. Interestingly, very consistent with her original conceptual model, she also saw the mobilization of iron and aluminum in solution associated with oxalic acid addition, suggesting again this pathway whereby oxalic acid may be mobilizing Mayon carbon and nitrogen. And indeed in this study, both of these separate pathways stimulated Mayon carbon and nitrogen priming, specifically from the Mayon pool. So then we followed this up in a recent study at the Kellogg Biological Station in a crop rotation gradient, going from corn to a corn, soy, wheat rotation with a rye cover crop. Not surprisingly, we saw both total carbon and nitrogen increasing with crop complexity, but here is the very finest fraction. It's less than 20 microns. This is the fine Mayon fraction. This is where we see most of the carbon and nitrogen gain along this rotation gradient at KBS. So most of the nitrogen gain is there in that Mayon fraction. These are sites that are not fertilized. We also saw along this gradient a shift in the microbial community structure. Again, changes in AMF, changes in actinobacteria and gram negatives, and a massive shift in potential enzyme activities with a switch in the carbon acquiring to oxidase ratio across this gradient. So again, crop complexity, shifting microbes, microbial investment, and carbon acquisition. And so how did this impact crop nitrogen uptake from soil organic matter and new sources? So this is the plant nitrogen concentration across rotation gradient in the greenhouse. These were not fertilized plots. You can see here that the this complex rotation had a much higher capacity to take up nitrogen and got a much higher nitrogen concentration. We added 15N clover residue to these soils to look at the nitrogen uptake of new versus old soil organic matter. And you can see in this more complex rotation, the plants are very happy getting the majority of their nitrogen from the existing SOM pools, the majority of which are Mayon nitrogen. We also saw much higher photosynthetic rates and productivity in those complex rotations. And so we saw overall the complex crop rotations increased Mayon and microbial community size. They increased uptake of nitrogen from soil organic matter nitrogen pools, reduced nitrogen uptake from new nitrogen inputs, and increased photosynthetic rates, stomata conductance, and intrinsic water use efficiency. Mayon, we think, provides a place to accumulate, retain, and recycle massive amounts of soil nitrogen. I think to optimize sustainable productivity, we have to continue examining how to build this pool, how to provide microbes and plants access to it, what are those mechanisms, and then how do we go about augmenting the internal recycling of nitrogen with supplemental fertilizer use. As this graph shows, what we're looking for is to have internal cycling of nitrogen, plants and microbes accessing that internal nitrogen and augmenting that pool just enough with external nitrogen additions to replace lost nitrogen. And with that, I'm just going to toss out this acknowledgement slide for all the people who actually did the work that I presented and turn it over to Johan and the panel. Thank you.
Thank you very much, Stuart. And actually, I want to thank again all of the presenters, the panel, because obviously it is quite a rich, presentation by everybody that probably stretch your mind from quite across a board, a bunch of different subjects, and I hope you understood most of it. But I see also that we indeed have quite a bit of questions coming in. And so I would like to invite actually now all of the panel members to put on their video so that we can have a discussion, with the audience. So thank you again. And I guess a first question that I see, and that was somewhere to be expected, is indeed that what are really the indicators of soil biodiversity that are valuable for farmers? And so I guess I would like to maybe invite Gerlinde and or Jonathan to give an answer to what are these indicators now that farmers can relate to.
And you then mean specifically the biology, or I mean, I guess the first one would be, but to a certain level, soil organic matter is quite related to soil microbial biomass, for example. But that's not a continuum. So that is when you are at the low end, that is a good indicator. Other things like pH can also really help. I mean, also when we look at papers from within fields, but also across fields, across the globe, pH is quite an important indicator in terms of community composition of microbial communities and even abundances. If we go beyond that, then, for example, if a farmer is afraid of a certain pest or disease, then they're also using, even in situ, qPCR type strips you can insert in your soil at certain locations and get a measure of what is the starting point of your level of disease, and do you need to be afraid there and choose another crop or something, or is it still below the threshold? So it will depend a bit on your specific question, and the ones I just mentioned now are still relatively general in relation to your specific crop.
Yeah. Yeah, thanks, Gerlinde. Maybe Jonathan, if you want to add something.
Yeah, I think, I mean, one of the major indicators that's often recommended are earthworms. I think the issue with earthworms is that they are seasonal, and so if you go out in the summer and it's very dry, don't expect to find large numbers of earthworms. So there's an environmental context to earthworm sampling. But if you are routinely monitoring your fields in the early spring, for example, when the conditions have been, moist for a reasonable period of time, you can do year-to-year surveys that begin to tell you if your soil is more active in earthworms. And I think earthworms are good from a farmer's point of view because they can see them. They generate large biopores, which are important, and they're important for cycling nutrients and also, as I say, improving soil structure. So I think they're a good initial indicator for a farmer because you can actually see it yourself in the field and you can make records. And if you have a standard protocol for extracting and measuring those, which you apply routinely, then I think that's great. And the other thing I would recommend is doing visual assessment of soil structure, because I think if your soils are well structured and the aggregates are coming together well, you've probably got the biology mostly right. And if the structure's bad, then there may be a number of reasons for that, but the chances are that the bad compacting structure and disaggregating structure will actually lead to poor biology, and you'll end up with pathogen problems, and you'll end up constraining some of the better organisms. So that's another way visually you can do things in the field without having to send off for lab tests.
Like rooting density, I thought that was also an interesting one that can be very visual, and that's where the food is going indeed.
Yeah, but that's a bit more tricky to measure, as we all know, the root density.
Yeah, but maybe if we build that up a bit with taking images and make a certain reference collection, I guess farmers also become more digital in that respect, and the comparative development.
Yeah, correct. I mean, I guess Sara and Stuart, if you have any others that you think would, from your studies that you think is a very good indicator.
Yeah, a brief comment on soil earthworms, which play a fundamental role in many agricultural systems, is that in arid and semi-arid systems, many times earthworms are almost absent. So in Southern Europe, for example, earthworms are absent from many agricultural systems. So in those systems, to have a measure of biological activity and other indicators could be used, which are probably more difficult to see more with the eye than earthworms, such as arthropods, beneficial nematodes, or enchytrates, or other organisms, even ants or other arthropods. But also in these systems, assessing visually soil structure might be very, 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 very useful. And of course, as Gerlinde said, organic matter, the amount of organic carbon in soil is, in my view, the best and the first indicator of soil fertility and soil structure in most systems.
I have
Go ahead.
Johan, I have a quick thought on this. I would say the most consistent indicator that we've seen, and this now extends back a number of years and over many, many studies, and it may just be the types of measurements that we make, is the ratio of enzymes that we would call carbon acquiring, like beta-glucosidase and cellulose degrading enzymes, to oxidases, things like phenyloxidase and peroxidase. And over and over again, we see as the soil becomes depleted, there's more microbial investment in the oxidase enzymes. And we could, talk at length about the mechanisms behind that, but we can save that for another day. But we do see that across lots of different cropping systems gradients now that we've studied.
Yeah. I mean, Sarah, actually, since you brought it up in dry land, you have these, so you have to think about some other soil indicators. But there's actually an interesting question also from the audience that is asking, like, which soil biota would you really like to improve in irrigated systems? So, you're under dry conditions, that's when you're using irrigation. And we know that irrigation is not always very beneficial. But so how do you do, your improvement of soil biota under those conditions? Any thoughts, or maybe somebody else, if Sarah is not ready to take on the question. But irrigated soils are an interesting one.
Yeah. I don't know if I understood the question is which soil biota do you want to increase in irrigated
Yes.
in irrigated systems? And I would say basically all of them. I mean, I don't know how should I choose one. So as we've seen, basically most soil organisms play beneficial roles on nutrient cycling, carbon sequestration, crop biomass, and crop yield. So, for example, soil animals, I've seen, for example, Johan, I think in your presentation, in one of your beautiful slides you showed as at the start, often soil animals are only shown as pests, so only arthropods and plant parasitic nematodes and so on are shown, while most soil animals are actually beneficial organisms. So, except pests and DC and vectors of soil diseases, I would say all soil organisms should be promoted in all agricultural systems. Doesn't matter if they are rain-fed or irrigated systems. The thing in irrigated systems, depending on the irrigation, is that the distribution of soil organisms can be very, very, very patchy, because can be very, very, very shaped by the irrigation type. So, but that is basically it. I would say all of them.
Yeah, thank you. Gerlinde, you seem to want to say something.
Yeah. Is it really just irrigation or irrigation together with some mulching or organic matter management? Because you still need to provide some food, right? So is the idea then that the food all comes from the crop itself and the turnover of roots? Or I just wonder, irrigation, is that just water, or does it also mean mineral nutrients and skipping the whole organic matter that contains nutrients? I think it's important to have that organic matter with kind of slow release nutrients and being a habitat and attracting like these ecosystem engineers. That water is a nice start, but I think you need more than just water.
Yeah. So there's a question also that, so until now we have been mostly talking about how how management or whatever is affecting specific groups. But there's a question from the audience also on how much do we have interactions, and specifically about how the soil macrofauna, how do they affect actually the soil microbiome and also the phytobiome. Any thoughts on that?
There have been, I've not done them, but there have been studies that have looked at these sorts of interactions. And it is clear that there are interactions between, for example, earthworms and mycorrhizal fungi, and they're not always as negative as you might expect. You might imagine that earthworms, by chewing their way through the soil, would break up mycorrhizal networks. But I think there's a kind of synergy between them that they tend to operate, as I say, synergistically. So, yeah, I mean, I think some of the bigger, more disturbing soil animals burrowing animals and so on, may actually cause adverse effects on some of the fungal structures and larger microbial communities and networks and so on. But actually, I think in well-balanced soils with a good diversity, these things tend to be compensated by better growth of the organisms anyway, and the improved porosity is often one of the key things, because I think we've often overlooked the importance of the relationship between soil structure and soil biology. and the extent to which soil biology plays a very important role in generating both aggregation and also pore spaces. And I think a really critical issue for maintaining healthy soils is that balance between parts of the soil which are being physically protected and parts of the soil into which gases and water can move in and out very, very quickly. And that provides really good functionality.
Yeah. So Jonathan, since you're on to the earthworms, there's actually some questions also from the audience about your systems, and there's a question about if there are also invasive or non-native earthworms that were present in those lays, or was it mostly native?
They're all native because we don't have there in those field systems, we didn't have any invasive species. So they're all native. What's interesting is, and I didn't really explain fully, the reason why we had pairs of lay strips was that out of each pair, one of the lay strips was connected right up to the hedgerow. So it was continuous to the hedgerow. And in the other of the pair, we had a barrier. It was a stainless steel mesh barrier, like a curtain that we ran into the soil. And we had a fallow zone inside that between the end of the lay strip and the field margin. And the idea was to see if we could prevent migration from the margin back into the field. Well, we saw no effect at all of the barriers. And we don't think that the barriers were being evaded. They were very long, actually, relative to the width of the strip. So sorry, it's a long answer, but the reason why I'm saying all this is that what we think was going on in those fields was we were re-recruiting from the depleted populations that were already there. So there were small numbers of earthworms, they were still there, of a variety of species. They didn't migrate in from the edge, but they recovered from within the field, which in some ways was encouraging. And we think the same was true with the mycorrhizal fungi, because again, there was no evidence that they'd come in from the edge, although it does have to be said that we did see some species of mycorrhizal fungi that are associated with the hedge starting to appear in the lays, but they appeared independently of distance from the hedge, and they appeared independently of the barriers that we'd inserted in the fallow zones we'd inserted. So we think they probably also were recruited from within the field, from very depleted populations.
So there's some people also that are interested actually on how do you effectively incorporate these lays into your crop rotation? Like what kind of duration do you want, and how often, you know, how long is the legacy effect of these lays? And so, yeah, what would be somewhere an ideal rotation with these lays is what people are asking.
Yeah, so again, I think it depends on what your starting point. So if you start with a soil which is very, very organic matter depleted and very, very intensively cultivated, you probably need a minimum of 3 years, because it takes quite a long time for the system to build the positive feedbacks. You probably don't want to run them for much more than 3 years, because you end up building up crane fly larvae and various other crop problem organisms. So there's and it's probably the reason why in the past, you know, the traditional rotation was a 3-year lay. But I think the one other thing I would say is, you know, I'm involved in projects now where we're not just looking at grass and clover, but we're looking at much more biodiverse herbal lays with specifically selected deep-rooted species, species that are anti-helminthic, so they're good for the livestock, species with a high tannin content, so they reduce the methane emissions from the livestock, and they also reduce the nitrous oxide emissions from urine inputs from the livestock. And so this then changes the system quite significantly, because you end up with much more carbon going in, bigger root systems, and in those cases you might get away with a 2-year lay, because they are supercharging the soil actually. But I think, again, it depends on your starting point. And some of the farms we're working on, they've got problems with blackgrass, which is herbicide resistant, and actually running the lay for 3 years plays a major role in depleting the blackgrass population. So you get those additional benefits alongside the soil quality benefits. And you asked me about at the end, how long, well, we're currently trialing second year post lay. First year post lay, we've got fantastic wheat yields using only half the normal nitrogen fertilizer. We were getting yields well above national average. So it just demonstrates the amount of fertility. And Stuart, you'll be glad to know, clearly a lot of that nitrogen's gone into the organic mineral fraction, and we're getting it back out again. So again, it does work in practice.
So actually, for Stuart, there was also a question about like which cover crops are the right cover crops in your rotations, and you know, to really build up that Mayon nitrogen. Any thoughts on that?
Yeah, I think when it comes to cover crops, that we should not let perfection be the enemy of the good, and we can always hope that we could have multi-species cover crops and increase the perenniality of our annual cropping systems, but economics dictate that that's just not often possible. Mm-hmm. And we've seen over and over again that the inclusion of any sort of cover crop is tremendously beneficial. So you have to remember, if you're growing something like rye, which is something I'm very familiar with in the types of Midwestern systems and Northeastern systems that I work in a lot, that oftentimes in the spring when that's incorporated before main crop planting, it may only be like this tall, right? It's very short, maybe it's this tall, but there's those small plants are below ground pumps, and they are sending far more carbon below ground than is suggested by that teeny amount of above ground growth. And we've seen this using isotopic labeling and tracking newly fixed CO2 below ground, that twice as much, as much root biomass is produced, there may be just as much in terms of exudates that are produced. And so I would just say, anything that you can do to get a cover on your soil at every time of the year, a growing plant that's pumping carbon below ground is going to make the microbes and the entire soil food web much happier than they would be without those plants growing there.
So there's actually also a question about, you know, so you've been talking mostly about a rotation, but what about polycultures? You know, would they have the same effect? So you know, intercropping or whatever. So do you expect the same as with these rotations? Mm-hmm.
Yeah, I just want to also make one comment that even those crops that are not fixing nitrogen, so take a grass, they are mopping up nitrogen that is otherwise vulnerable to loss. And that when we incorporate that crop residue, that cover crop residue, that's nitrogen that's being restored to the organic cycle and subsequently made available for crop nitrogen use. So we shouldn't just think of the benefits of legumes to nitrogen, but also anything that's in there that's retaining nitrogen in the system and recycling it so that it can cycle back through the organic pool and made available again to plants and microbes. I think incorporating perenniality is great when it's possible. There are certainly a huge number of benefits in terms of below ground biomass and the building of soil structure that others have talked about associated with perennials. If that's a realistic scenario in your cropping system, go for it.
So there's also two questions or a few questions from the audience about, you know, you're talking about these ways of these new practices or whatever, but what is the you know, the economics associated with that? And so from an economic perspective, what is the most efficient way of improving your soil biodiversity?
I think that depends on your economic system. So for example, in the UK, where, you know, the government is now providing financial support directly to improve soil quality, then some of these cropping systems with lays become financially viable in a way that they won't in a system where there is no direct support for that kind of intervention. But I would also say that, you know, that there have been a number of studies now that have demonstrated, for example, for livestock grazing, the traditional model that was used in many Western European countries of growing ryegrass, heavily fertilized to maximize production of sheep and cattle, has subsequently proven to be economically far less advantageous and environmentally far more damaging than using mixed species incorporating legumes, which provide better feed quality, reduce the greenhouse gas emissions, and massively reduce the nitrogen fertilizer requirements. So I think we're in a changing world at the moment. The price of fertilizers, particularly nitrogen fertilizer, is also having to causing many farmers to have to rethink high input, high output systems, which have got low efficiency. And the one final thing I'll say in relation to this, again, coming back to the issue about perennials and perennial break crops, particularly in systems using reduced tillage, which of course saves fuel and diesel costs. If you're moving into a system with reduced tillage, having previously grown a really deep rooting ley, for example, it opens up the pore space deep in the soil for the root systems of the following crops to go down and gain access to nutrients much deeper in the soil than they would ordinarily. So then you suddenly improve your nutrient use efficiency and water use efficiency of your follow-on crop, even if you use a very reduced tillage system that might otherwise not be able to deal so well with soil compaction. So having those living root systems actually doing the burrowing and taking out the steel and the diesel and using the organisms to do the hard, heavy lifting for us strikes me as being one way forward that actually we perhaps need to try more widely, because where we've been trying it, it works, and the results speak for themselves in terms of fertilizer savings and also really good crop yields as well, at least for a couple of years.
Thanks. So there's an, okay, Gerlinde, you want
yeah,
to
I was
say?
just going to add that maybe also irrigation savings. I don't know whether they irrigate a lot, but like with drier summers and springs, I mean, yeah, having those roots go deep enough and having the sponge activity, the buffering, because of the well-developed soil, yeah, that plays into risk reduction, I think.
Yeah. There's also a question actually on another aspect of the economics, but then more from a research perspective. There's the indication that you know, you guys are all having quite some funding compared to other places in the world, and so they're asking about, you know, what are some of the more economic ways of assessing, you know, the soil biodiversity and how it's impacted by management in agriculture. Any thoughts on that? What would be, you know, your preferred, not so expensive methods to do some good assessments?
I don't
Sarah,
think
maybe
that
you
should
have,
be,
yes.
yeah, I don't think that should be very, very, very expensive. I mean, everything costs money, of course, but in terms of soil diversity and soil organisms and basic indicators, with basic lab equipment, many of those things can be performed without going into qPCR or metagenomics or very expensive systems. For example, it comes to my mind the tea bag initiative, in which tea bags are used to measure organic matter decomposition worldwide. That is a well-established methodology to measure organic matter decomposition, which is closely linked to nutrient cycling, using only tea bags. And with basic lab equipment, like a very simple microscope or a binocular, many organisms, many soil organisms can be assessed. So I don't think that is that expensive. I think you might have good quality data with basic lab equipment. At least I am used to do it because I work with morphometrics. So I don't spend thousands of euros in molecular techniques. I just use the basic microscope.
Sarah, while you're talking, I wanted to also ask another question that's in there, and that's about you know, when you show the relationship between the nematodes and the crop yield, that this positive relationship, where you also, I mean, was that the whole population, or was it, and did it include plant feeding nematodes, or was it a specific group of nematodes that was showing that positive impact?
In general, soil organisms abundance is not the best indicator, because that might include very different organisms. Some of those might be beneficial, some of those might be negative. So you have to go a little deeper into the community you are studying, and at least being able to differ between what can be seen as beneficial organisms and harmful organisms. In any case, in the case of nematodes, in general, beneficial nematodes are always positively correlated to crop yield, but we know well a specific group of soil nematodes which are basically bacterivores and which maintain a very close relationship with soil microbes. So by grazing on soil microbes, they promote nutrient mineralization. So more nitrogen, more mineral nitrogen is available in the soil, and soil fertility clearly increases. So there are many studies in different countries in which we've clearly seen that the more those bacterivore beneficial nematodes you have in your soil, the higher the crop yield is.
Mhm. There's also questions from the audience about, you've been talking quite a bit about these crop rotations. We have the lays, we have maybe the crops with deeper roots, but there's also questions about, what does compost, for example, do? And also what chemical fertilizer can do, and is it only negative, or are there also positive effects of fertilizer, especially then when it comes to, for example, the Mayon N, can you have some of that being put stabilizing by chemical fertilizer, or this compost, or other amendments? Maybe first on for Stuart, the effect on Mayon N. What do you think?
Well, one way or another, we have to replace the nitrogen that we're exporting from our agroecosystems. We have very, very large, in many systems, very large internal supplies that nitrogen is cycling and potentially made available to plants and microbes, and we could certainly be doing a much better job intercepting nitrogen, having our plants intercept nitrogen when it's available. That's an area that we are very keen to learn more about. I think that modest nitrogen applications and what those mean are going to be system dependent. I think that they can support this process of accumulation of nitrogen in the Mayon pool. Jonathan touched on something earlier, which we know well, which is that a lot of externally applied nitrogen is going through that organic pool anyway. It's entering into the Mayon pool and subsequently being made available to plants. And that's why the fact that that whole process of external nitrogen being taken up by microbes as they decompose other organic matter, entering the Mayon pool, cycling on and off minerals, being remineralized, and subsequently being intercepted by plants is so important to better understand. Right now we just black box it, and we just come up with these general estimates that, some very small percentage of soil organic matter and it's going to be available each year, but our laboratory methods and crude assessments are in no way good indicators of what's happening in the rhizosphere and the potential for plants to take that nitrogen up. So to summarize, I think that external nitrogen fertilizers can augment this process, not suppress it, but we have to better understand the processes to optimize the two working together.
Yeah. Maybe as a short follow-up question that's been asked to Jonathan is the effect of nitrogen on the AMF in agricultural systems.
I don't know, to be absolutely honest with you, but I don't think there's a massive effect. And the reason why I say that is that, for example, with wheat, if you change the management but still use the same nitrogen fertilizer loadings, you still get quite high levels of mycorrhization. So I think the issue with a lot of these crops is that mycorrhizas are phosphorus sensitive, but they're less sensitive to nitrogen because the arbuscular mycorrhiza is adapted primarily, but not exclusively, but primarily as a enhanced efficiency mechanism for the capture of phosphorus. And their role in the uptake of nitrogen is much more uncertain, to be honest with you. I mean, they may operate in organic systems and systems of low input where mycorrhizals may be involved in recycling organic nitrogen. But in high input systems, they probably play a minimal role. I think the other issue that's quite interesting is the extent to which some of the chemicals applied in agriculture impact mycorrhiza inadvertently. And there's been quite a lot of studies on things like glyphosate, where glyphosate additions do seem to have negative effects on mycorrhizas under some circumstances. But it's interesting, in our experiments with leys, we've sprayed off the leys and direct drilled, and we've still got better mycorrhization than we have in ploughed or in shallow disc cultivated land. So it does suggest we don't have a control with no glyphosate, but we still have very good mycorrhization after use of glyphosate with a direct drilling approach into ley.
Mm-hmm. Actually, still related to this nitrogen, Stuart, there's a question about when you want to really study that role of nitrogen and the nitrogen mineralization potential in Mayon, if there is a specific fractionation method that is best, or yeah, any recommendations on that?
Well, of course, this is something soil scientists love to argue about. Can't get enough of at times. I think that the simplest approach is just to use a simple soap like hexametaphosphate, shake the soil, pour it through 53 micron sieve, and that separates your less than 53 microns, so silt and clay fraction, from the sand fraction, which would include the palm. So that particular organic matter, by definition, is ending up in the greater than 53 micron fraction. Now, when we get into more detailed mechanistic studies, in my lab, we're very interested in separating out that less than 20 micron fraction. And the reason for that is that the 20 to 53 micron fraction captures the core silts, and we think a lot of those are primary particles, and they are not associating with organic matter in the same way. They're acting more like a primary particle, just a small sand. They don't have the surface area to volume ratio in the reactivity, so we get very interested in that smaller Mayon fraction as sort of a hotbed for nitrogen accumulation. But again, that takes quite a bit more work. And so it depends on the study and what sort of resources you have available. But if you remember back to that study from KBS that Bowles et al. Did, it was in that less than 20 micron fraction where we saw most of the accumulation of organic matter associated with crop rotations and nitrogen in particular. Mm-hmm.
So there's also a question that's maybe a bit critical for you, Stuart, and is on the so you're clearly focusing on the Mayon and how that plays a role in providing nitrogen to the plant. But what about the POM fraction? Is it really just not playing any role, or is it just less, or what's your thought about that?
Well, I think we should be careful about getting overly focused on any one fraction. They work together, they interact. The breakdown of one supports the other. That POM fraction is a really rich source of carbon. It has a much higher carbon to nitrogen ratio. It decomposes relatively quickly. In agricultural systems, a lot of microbes we tend to think of as carbon starved. These are carbon limited systems, and that POM provides a real important source of carbon. It serves as a seed for aggregation. It serves as a seed for the formation of microbial biofilms. It's the decomposition of POM that forms Mayon. In turn, the Mayon nitrogen may help support the decomposition of carbon or of nitrogen limited POM. So there's important interactions between these different pools, and they are both serving really important functions in the soil. You could, coming back to aggregation, you can think of the Mayon carbon and nitrogen and that pool of nitrogen helping to support the formation of little aggregates. And then it's the palm that helps to, and the formation of microbial goos and things around it that helps to bring together those small aggregates forming around Mayon to form the types of bigger aggregates that really increase pore spaces. So all the organic matter in soils is super important. We are just focused on Mayon because it's largely been overlooked as a nitrogen source. It's been considered somewhat inert. And we think that there are these plant microbial mechanisms to access this pool that's like 80 to 90% of the total N.
So we're actually been talking quite a bit about, just how these microbes and then how they turn over nutrients and lead to increases in plant uptake and such. But there's actually also a good question, and this was directed actually to Gerlinde about if you're talking about, the food security and how microbes can play a role for food security, are you really only talking about the yield, so the quantity, or is it also the quantity and the quality? And so are there any indications of how microbes can actually play a role there, in improving the quality of the product that we're having?
Very good point. Definitely quality. I think we're in the past, maybe some people still have been focusing a lot on this quantity, but it's definitely not only carbs that we need a very balanced diet. And so also the soil needs a balanced diet. And when we think of these closing loops and recycling, we also need to take care of good balances and avoiding pollutants. When it comes to things like micronutrients, I know, for example, that mycorrhizal fungi, they really can be very beneficial in that for helping plants to acquire the poorly available, but still present. They need to be present. That's, of course, a prerequisite. If they're not present, it's not that any of these organisms are fixing it from the air. That's quite different from nitrogen. So they need to be present, but promoting accessibility for plants, yes, the microbes have a big role in that to play in terms of zinc and many other micronutrients. I think there's also a role for really taking a good look at the roots themselves, like in our breeding efforts for new crop varieties. I think we should be more aware of it. And even also crop genotype or crop species combinations that via their exudates promote certain, via microbes, often even the availability of certain micronutrients. And then actually we also work on one of these examples, but maybe it's a bit an exotic one, where we also work with traditionally fermented foods. So basically actually augmenting the quality of the food product. In this case, it's a fermented grain-based beverage, non-alcoholic, and the inoculant that really provides a microorganisms and also some enzymes or roots harvested from endemic plants. So that's one of the other projects we're running, just to see how is this relation between these microbial communities coming from the soil into the roots, into this beverage, into even the vitamin compound. So yeah, so there are links out there. Maybe they're not always that obvious, and maybe it's especially more traditional type of foods, because here we, like, at least what I experienced in the Netherlands, we try to kill things to protect it from not being risky for us to eat. Although I like some raw-based milk type of things, but they're quite rare here. Just to say, yes, there are direct connections, and quality is super important. And I think that's more on the agenda now than the quantity. Of course, we needed to have that quantity fixed first, I imagine. But also thinking of overuse and micronutrient deficiencies that really occur. Yeah, they're more difficult to study, but they're very important. Yeah, there was a bit of a long answer.
Maybe, thanks. Maybe just one last question. We've been talking about this compost, the rotations and whatever, but there's actually an interesting question. So what's the role of livestock in all of this? And can livestock actually help with building up soil biodiversity, building up soil structure, having a good soil that harbors indeed the soil biodiversity? So livestock.
So we're running experiments now comparing lays managed by mowing versus grazing. And there are slight differences, but they're not that great actually. And I would say it isn't essential that you have livestock there. And there are disadvantages with livestock. You can get surface soil compaction, you get localized problems with livestock causing soil erosion in a way that you don't get if you mow. On the other hand, you get the manure and the urine nitrogen cycling coming back. On the other hand, you could potentially add those materials back from livestock that's housed. So you don't have to have the livestock on the fields necessarily. You could produce silage and feed it to the livestock and then inject slurry back into the ground and provide some of the nutrient recycling in those routes. So I think, you know, the upshot is I don't think you absolutely have to have livestock. I think the systems with livestock are very efficient, and they can be made very efficient by choosing species that give very good feed quality, suppress intestinal parasites, and also play a role in reducing methane emissions. But as I say, I don't think the livestock are absolutely essential. Even in regenerative systems, it's possible to generate regenerative farming systems that are livestock free, I think.
Yeah. Yeah, but obviously we do know that in organic farming, I mean, there's a lot of manure being used, and organic farming, I mean, is one of the ways that we see that life is much more in the soil, and, you know, because of that input. And obviously, you know, if you don't have the livestock to provide that manure, then yeah, what are you going to replace it with? You know, I mean, obviously you can do some lays, but Jonathan, you're off.
Sorry, living root carbon is the key thing. Yeah. Living root carbon, and that's what's missing from a lot of our systems actually, because our crops have been bred to have relatively small root systems, or indeed we're actually cropping the roots in the case of potatoes or sugar beet. So,
but then
okay.
when it comes to lays, I mean, you do mow them, right? And you also want something to eat that, and then to produce poo with structure, hopefully, like farmyard manure can be really beneficial in helping to build soil organic matter, which contains carbon and nitrogen, at least, yeah, from the Yorkshire Dales experiment I was involved in. The farmyard manure, that one had a really big impact, positive. But of course, you need a certain, you shouldn't overdo it either, but in such a way that your plant community can be maintained. And like in this diagram that I showed, the Van Zanten diagram on this circular agronomy, actually, especially livestock that feeds on grassland type systems that we can't feed ourselves on, those are seen as quite crucial. But of course, they should reduce their methane emission and nitrous oxide as well. So that's something we're going to work on.
I'm very sorry, we have 1 minute left, and so I want to thank everybody, especially the audience, but also you, obviously, the panel, to keep the discussion going. And I just want to say to the audience that indeed this is being recorded, and it will become available via YouTube. You will get an email about that, if I'm not mistaken. So you can always go back to the recording. And so with that, I would like to close it off and wish you all a good evening or the rest of the day, depending on where you're at, or a good night. Yeah, thank you all.
Thank you very much.
Bye.
Thank you, everyone.